particular research. And also publish standards, courses, there are more new courses, in fields from conferences, like also ebooks from well, so we have actually this library available. We even have available on the IEEE Explore platform. So if you return to the IEEE Explore platform, you'll be able to search all of this immediately. And also, not only this, we also host the other publishers as well. IBM has two journals, right? As I mentioned in MIT in the past. So, I think a quick slide which I would like to show to all of you. You can see most of you are sure that in the Iron Man movie, so use it to say that you and an Iron Man actually information. You can see here, if you're working on something, and if you use this particular keyword, that is in phrase format, you will get more than 168,000 articles. So, now if you use water system, water system, laser technology, potential system, there are so many articles available in IEEE. And for example, if you're working on control systems and you have some idea in your mind to see that you have to spend very less time in researching and more time in reading to what you should be doing to respond to all the objectives help you and features very much effectively. Also, to quickly highlight some enhancements. So, going down, quickly. So every year, these new journals and new teams, and this last year, we are expanding to the expo. And this year, we have 6 more journals on cloud computing, and that you can get the topics in power electronics, new science, and remote science. Translated to this year, these have been launched, and all the it's like public. You have to launched in the month of May, and it covers all the fields covered by IEEE. And just to quickly go to the platform, we explore the digital library platform. Here is what's popular. If you'd like to know which of those keywords people use heavily when they come to IEEE Explore, here. And if you'd like to those articles that are on the basis of them, I think it is published. So for IEEE Explore, you can come here. So last subscription was July 1st, and we had 11 issues and the conference. So they are the latest, fresh content which are available onto the IEEE Explore platform. So this is the basis. So we will take a quick the type ahead to as per your target. We can see here these are 2020 again. Well, they're all up to 2. Although pretty much important is having researchable knowledge skills under to send more to reading your. So always up to your top. Even this would like to see the articles which are just published. We'd like to know those papers highly cited which are referred heavily this and the left hand, a lot of top down the result. So, you can use it not to refine it and to and company of two. Well, so only articles we publish two articles already it's but it's a source that will be published in future, so you have an access as well. And right hand side is not this block is also given back to I. So, not even except for it is IP authenticated. Wherever you are within campus, read this. So coming down, not for it, and search for particular app, look for authored paper about this particular institute. Your country, you can see. Coming down, application title, focus your art. Coming up, you can use it and to look for publishers like IEEE, IEEE, IBM. Also, let's use a good keyword here, energy harvest. Yes, Prof, you have some questions to me? Okay, I understand. I'm not sure. Okay, let me please suggest what should I'm not sure if phone, mobile phone, the phone as near as possible. How is it now possible? So it should be fine. Here the person is once you use for keywords, like one just and do that and take for example so this is how you can go on. Okay, now I will probably cannot. Unless a problem, you can go on here and I think offers personal file. So okay, just I'm not sure what's happening. I'm so sorry for this. I know that technically there might be some problem. So please suggest what.
Hello.
absolutely free. Just get into your profile and then get started.
We'll just cut short this presentation to 5 more minutes.
Get into your profile. There are some quick things to note about. One,
We'll probably share this presentation and
you can
just see the
and set alert for the search. And then
for probably searching.
For example, if you're interested only in these two articles, and you don't have time today to read them, you can save to project folder. So I typically in 2 months save the project.
I've asked him to cut down the presentation, please, like just 5 more minutes of this.
As many and then save. So these two articles have been moved to the project folder. So in future, we will be able to add into my project. So your search will not be lost, your article will not be lost here. So you just come here and then click on wireless and select it. You should be able to do it here. Also recently, I actually came up with HTML version of the article. The article has a separate to read the article in much more effective manner. Help of the HTML space. So you just need to click on author references of it. And if you are logged into the IEEE so you sign in, you can even read the text just quickly navigate to that. And we have the advanced search aspect. Combination of searches. So for example, if you're looking for RFID and the back, here you can restrict your search on it and just to conferences, journals, etc. and then search it using that phrase and then get a 115 result. If you go down again, you will be able to see that RFID and mobile phone are not at all related. It means to say that they are no more near to each other. But in that case, you can answer. Very powerful. I use it. I want to command search. Very for the operator to use the same. So for example, you are looking for RFID and mobile phone related article, has very should be separated by not more than 5 words. You can use near /5 in the operator, you get a word result. So wherever you go, you will see that these two are like almost related. So the number is up to you. You can go on. If someone operates, and you can see you get one. So that's the beauty of the search. We always provide operator guidelines and also do that. This is the last part of the session. I'm sorry, I want to show you. Maybe this one I will just quickly show you because recently we launched the once you start browsing the journal, the browse journal page has lots of information which you can for example, this particular journal, I'll just take the present effective computing. And see the impact factors. So coming homepage, there's IEEE training and tools. Once you click on the training and tools, in future, if you'd like to attend my training session, Here you can sign up for one of the courses. My next training session is on 1st August, 2nd September, and 3rd October. You can join any of them in the future and get you the 1 hour session. Is what there are self-paced tutorials available in the IEEE Explore client service page where we can listen to short audios and various features. Very frequently asked questions, very important to most of you. I would like to see the author resources. What kind of author resources are available to me? The facts for authors, what are the author tools, publication policies, submission guidelines, each and everything is available to all of you here. That will happen in the article submission stage, post acceptance, etc. So to event, you would like to see what are the cloud in India. Still the cloud. So I just choose that. And see, there are 9 conferences in cloud, so you can even want to plan attending it. Or if it's sufficient time to set your paper, you can also start looking to some of the people. So, Prof quickly asked me to quickly help with the membership benefits. On the membership and services page, you have a lot of member benefits available. Here is the career phase. Maybe if you're a grad student, you will get various benefits. Not really tangible, but yes, you will get the access to the IEEE spectrum from the latest magazine. You have access to some of the e-class, the class you would separate. You have access to other world. You have copy of the magazine. Whenever there is a night, you will be invited. You can make a lot of people definitely there. And then also you can choose the types of vendor contact numbers we have. So the speaker to like what we can various so thank you very much for your time. Thanks to Dharanjan. All of you actually thanks to from grants to organize and to brief session where I hope this session has helped you. Not much weight, but at least highlighted
Of Mr. Dhanu. So if any of
not known. Here are my contact details.
or IEEE
IEEE@IEEE.org, and here is my phone number. And if you would like to organize similar session webinar to your institute, please feel free to write to me. And also, if you would like to have access to IEEE sessions, you can write to me. So next time again, we would like to
all right. Yeah, it's very sorry for that botched up presentation. The entire problem was with the audio, and we couldn't get the entire audio conferencing set up properly. Uh, we had asked the IEEE client services manager to forward the presentation anyhow, so that some of you who want to go through these member benefits as well as his entire presentation on how you can really make use of IEEE Explore and its advanced features. So you can have access to that if you want. We'll no, we'll just start off with the next session right away, and if you have any more questions or if you want to get in touch with Mr. Dhanu, you can take the contact details either from Debanjan or myself.
আমরা পরে ভুলে যাবো ওদের কোন কথা কোথা থেকে এসেছিল। নেট তো লাগবে না, বাস তাহলে আর কিছু প্রজেক্টটা লাগাবার ইয়ে কোথায়? পেছনে কোনটা ছিল সেটা তো বুঝতে পারছি না। এটা তো নয় মনে হচ্ছে, এটা একটু ঘোরা দেখাচ্ছি, এরকম রাখতে হবে। স্যারকে কলার একটা দিয়ে দাও। ওয়ান্টেড আছে তো? ওয়ান্টেডে সার্চ করে দেখুন না। পাঁচ হয়ে গেছে।
Okay, good afternoon. So So I shall be talking about that microfluidics in biology basically, not in general. First I shall start in general things, then I shall slowly switch over to biology. Basically, maximum application in microfluidics nowadays is coming in the biological field. Okay, now first we come, what is mean by the microfluidics? That you are MEMS, everything you are coming on. What do you mean by the microfluidics? So generally we are thinking that fluid flow in very small quantity or small situation. Like say if fluid passes through a tube, generally every day we are seeing your that pipe, water is passing through the pipe and everything. But if same fluid is passing through a channel of dimension around say micrometer range, around say 40 micrometer or 100 micrometer, then what will the behavior changing? Okay, first question. Now why you want to study that one? Why we shall study the fluid flow in a micro situation, like say micro or nano situation, and what is the basic physics, chemistry behind it, then what are the applications of it, that I shall be coming it out. Okay. So in general what is happening, when fluid flow in that micro size, then what is happening, that it becomes your laminar flow, not turbulent flow, and that makes a lot of things in the coming around basically. And as that feature is decreasing, your surface to volume ratio is increasing. That makes a lot of phenomenon is coming, which is not generally we are not seeing in large scale flow situation. If we come to biology, then where it is we are seeing every day. You are seeing every day, but you are not visualizing that what microflow is going on. So like say in our body itself, what is the cell dimension? Each cell we are composed of say 10 to the 9 cells. 10 to the 9 to 10 to the 10 cells in our body. So each cell dimension around say 10 micrometer. 10 micrometer. Like say RBC, if you can think about the RBC, it is a dimension around 9 to 10 micrometer. It is flowing through your blood vessel, then slowly going to capillary. Capillary dimension is around say 15 micrometer. Now if you look for that whole pipe, slowly it is going to your small one. Then what is happening to that your RBC or blood cell, and what is happening to the flow? And again you think that in the cell itself, interstitial flow means one cell to another cell, how the nutrients are passing, their velocity is around say around 0.2 to 1 micrometer per second. 0.12 to 1 micrometer per second your linear velocity. So in that fluid flow, what is happening to the cell, and if the fluid flow is increasing, what will happen? So these are very basic things. Then application things is that with this biophysical means nature, we can discover the new phenomenon also, that I shall be telling. Again, if you come across about say nowadays one term is coming, lab on a chip. Lab on a chip means you have a very miniature chip like say 1 centimeter by 0.5 centimeter diameter. In that chip you can do almost 100 chemical reactions. Like say every day what you are doing, you are taking a blood, giving to the clinics, they are taking say 10 tests in 10 tests. Instead, with this microfluidic and MEMS platform, we can perform almost, you can tell that all the general blood tests within a span of might be a half an hour things. We can finish up all the things. So you are thinking it is a dream, it is not dream. It is coming to the picture, and in our country maybe coming couple of years from now, basically it will be coming in the picture. So this is another part. And now if you come to that pharma industry, so they are utilizing billions of dollars or rupees for the discovery of drugs. What we are doing, we have maybe say a library of 10 to the 9 chemicals, we are testing say 1,000 tests. So now if you think that how much time it will require to test all these cover, but instead if we make a chip with a high throughput means readout, which is possible by fluidics way, then we can perform these experiments. So with these three concepts, like say biochemical reaction in a chip with a view to diagnostic development, then some cell behavior, single cell behavior in a fluidic situation, what is happening, like say cancer cell, normal cell, what is happening, that is impossible without fluidic platform. In the macro platform, you cannot unravel that what is happening to that single cell level. So those things I shall be basically discussing. Okay. So these are normal things what we discussed so far, mentioning that example in nature is human body in oxygen transport system, blood transport system in say capillaries, where that fluidic behavior, microfluidic behavior is really going on, and that is laminar flow, not turbulent flow, and that makes a lot of things. That is your laminar flow. Now, as I mentioned that as you are decreasing that feature, your surface to volume ratio is increasing. So that becomes a microscale flow is equal to laminar flow. Laminar flow allows control mixing. Like say if you make a red means your fluid color red, another fluid color is blue. If you pass through in a fluidic channel, they will mix up only in the interface. Nothing will happen to the mix. But if you increase the diameter of the tube, they will mix up. So that is one of the benefit, or you can tell that it is one of the drawbacks also, because every system has a positive and negative effect basically. Okay. That I told, oh sorry, it is going back basically. Okay. Now what are the forces responsible for your microfluidic flow and how we can perform these fluidic experiments? Now if you look for that in a tube fluid, how fluid is flowing through your pressure driven flow, you are giving high pressure and low pressure, and with this flow. Same thing here we can do it. So one is your, everybody is doing that pressure gradient, pressure driven flow. Then another is your capillary effects. Every day we are saying that if you have a blotting paper, if you soak it, liquid is going out, going top in the due to the capillary action. That is capillary force. Sorry, capillary force. Then by electrical force we can also driven, because that fluid is always has some charged. So with this charge we can flow by electroosmosis, dielectrophoresis we can actuate. Then magnetic force, then rotational force, means if you make a like a CD disc, that rotational, your centrifugal force. With this force also we can actuate the fluid at that microliter dimension. Then we can use sound, that is acoustic stream. If you can use the sound to actuate the liquid at the micro scale, then it will be amazing. We can use the light also to actuate the liquid, because light will influence the surface tension of the liquid, and with this help we can actuate the liquid at the microliter level. Okay. Now as I mentioned earlier, that used for analyzing thousands of samples at once, means high throughput way. So like that, this is the normal way you are doing, traditional way lab, you are doing say 10 chemical reaction or biochemical reaction, 10 tubes, it takes might be a couple of hours, even a day. But same thing we can do in a fluidic platform, dispense say maybe 5 microliter, hold the sample, we can disperse in 10 droplets and do the chemical reaction and perform the readouts. So that is the beauty of that microfluidic platform for your diagnostic development. Means so like say blood glucose level, urea, etc. Every day we are measuring, means with the help of clinicians and going to the clinics. But you have not to go to clinics. You just prick 1 drop of blood, give it, and it will give the readout of everything. So that is that things are coming around. So microfluidics we can use where that in a situation where sample volume is very less. You cannot get sample more than say 5 microliter. Where time is constant, you want to get that data within a say maybe couple of minutes or couple of hours, not days in terms of days. And you can use this platform where the resource limited situation, because where is not high gauged instrument is not there. In the total, the grassroots level, you can do that experiment, or you can perform the test to get the results basically. So we can, that can be used for that medical diagnosis, biochemical reactions, molecular and cell biology, particularly for say, if you want to say at the chemical reaction at the single cell level or single bacteria level. I think if you have a little bit biology knowledge, you know that when you are doing the biochemical reactions or microbial thing, you are taking a population. Say 10,000 cells you are doing the reactions. But here instead, you can take a single cell and do the reaction. At the single cell level, you can get the information. That we can perform with that fluidic platform. Okay. straight away I shall go to that high throughput medical device and at low cost. What we are doing, I shall be telling here. Here, just it is one of the example that DNA hybridization that might be you will be knowing or you may not be knowing. That doesn't matter. So any molecular level, diagnostic level nowadays is coming. DNA hybridization means if it is some mutant part of your DNA is really responsible for the disease, doctors or biochemist or clinician, they want to know that whether mutation is there or not. Like I am giving one example is malaria. So malaria is nowadays rampant, and even that multi-drug resistant malaria is rampant. Now, doctor want to diagnose that malaria within a couple of hours, not in terms of days. Then he can diagnose it. So that can be done by the molecular ways. So we know that what part of the DNA going means mutated. How it can be done within a couple of hours. Generally, you are doing it takes 3 days for the hybridization, very diffusion way. But can we do perform that reactions, get the readout within a couple of hours? Think is that very simple. So this is that one part of the DNA, one strand of the DNA, complementary strand will bind and it will hybridize. That is the normal biology. But where fluidics is coming? Fluidics is coming at all the molecules of the dimension at the level of say angstrom level. And we are doing the reactions at the microliter level, and we are giving a flow. Generally, all the reactions, if you give one drop of say blue ink in a glass, what will happen? They will diffuse. Okay. So diffusion takes a lot of time, and diffusion depends upon the how much distance they traverse. But here, as distance is very less, reaction will be very quick. And again, that micro confinement effect. All the things are going on at the microliter to nanoliter level. So in the micro confinement effect, as well as that is your fluid velocity, means convective flow and diffusive flow, both are helping to get the molecules coming closer together. That is why you are getting fast reaction. It is not that we can increase that chemical equilibrium constant. It is impossible. It is a physical phenomenon. Only what we are doing it here by manipulating that your dimension of the vessel, getting the molecule very close together, by which they can interact each other very quickly. That is kinetics we can change. We cannot change the delta G or chemical reaction we cannot change, but we are changing that fluidic behavior with the help of fluidics, that DNA hybridization we can do very quickly. Means basically you can do within 15 minutes. But if you can do normal traditional way, it takes at least 8 to 12 hours. 8 to 12 hours. Okay, that is same thing. Now, if you look for that, how that flow is going on, if you go through your pressure driven flow, that is your parabolic flow profile that will be coming on. Here is the maximum velocity, and this is the normal things actually. And that channels, generally we are doing the reactions in a 3 centimeter length and 500 micrometer width, 50 micrometer height channel, all those reactions generally you are doing. But you can decrease that feature to get that reaction much better way or faster way. And this is your electrokinetic behavior. If we actuate that by electrical force, then that liquid front is going without your parabolic way, it is straight way it is going on in that way. And as I am telling that mixing is a problem, means when in a laminar flow 2 liquids are going on, they are mixing at the interface. But can we mix it? So it can be done by using both pressure driven, that is your serpentine channel, or we are using that pressure driven flow as well as electroosmosis to mix up that reactions also. So you can further speed up the reaction by a order of 10, we can increase the reaction rate. So this is an example where we are focusing it here. Say readout is your same array. We are spotting micro arrays 50 micrometer diameter in a 1 centimeter length. So this is your maybe target DNA we are spotting. Then we are giving that is your analyzing DNA, which has a probe, basically red dye, and that will bind the target DNA and give a red color. A very simple things. So the readout is your red spots. Now think is that how fast we can do that reaction with the help of a microfluidic channel with a particular flow rate. So that we have observed that you can get that is your time at the rate of say within a 30 minutes you can get near almost plateau. Means within 30 minutes with the flow rate of, here flow rate is not given anyway, that is yeah, 20 microliter per minute, and that channel dimension is 3 centimeter length, 500 micrometer width, and 50 micrometer height. Here, if we go the flow rate of 2 microliter per minute with your match probe, you will be getting this color, and mismatch, they will not binding, they are not getting color. So you are getting that readout within a say 30 minutes. Why you are getting 30 minutes? Because here both diffusive and convective flow is playing the role, not that only diffusion. If we do only diffusion, it will take maybe 5 hours to get that much of readout, that much of color. Means if you do the reaction in a simple in a platform like in your glass plate or everything, that much color will not be getting if you wait for even 12 hours. You will not be getting color, and you will be getting color maybe after 12 hours, like this type of color will be getting. Means with the help of this phenomenon, we can decrease that time for a readout. And basic cause is that we are not changing that is your equilibrium constant between the binding of two DNA strands. We are changing the kinetic rate only, forward rate constant and your backward rate. That we can change. That we are coming to in protein-protein interaction more better way. So that is in case of malaria, we are giving that we have already done. Just give an example. So in a nutshell, for DNA hybridization, what we can tell that rapid DNA hybridization is a key requirement for on-site and high throughput detection and diagnosis in several diseases. So what we can do it, this is only one phenomenon that you can multiplex it. In a one platform, you can put 10, 20, 30, even 40 things. So within a one platform, within the same time period, we can get 40 readout. That will be showing later on. Conventional device which exploit the DNA hybridization are of passive type. So DNA hybridization occurs slowly by diffusion and hence suffer from various limits. That generally what you are doing. If we do the same reactions in your glass plate, you will not be getting that much of signal. That because only due to the only diffusion. In the microfluidic allows active micro devices in which the fluid flow can be employed as a controlling parameter for the obtaining faster rates of DNA hybridization. So both convective and diffusive flow is playing the role. And again, for increasing the mixing, there are a lot of different features we can incorporate also here. So within microfluidic platform, maximum binding occurs within 30 minutes, as I saw in the earlier slide, and you are getting. So that is that means you can tell that a benefit, or you can tell that achievement of that fluidic platform for DNA hybridization. So next we are coming, as we are mentioning that in which what are the forces by which we can actuate the fluid in a microliter situation. One force is pressure driven flow, that what we are so far discussed. Another force is centrifugal force, like your CD platform or CD. Everybody in the compact disc, you are hearing that your compact disc, you are rotating by centrifugal force. We are utilizing the centrifugal force to actuate the fluid in a microliter situation. So here, what are the forces? The centrifugal force, this is Coriolis force, this is your drag force, then this is your, that is viscous drag, and this is your surface tension. And if we derivatize that centrifugal force and Coriolis force, another one force will, that is not mentioned here, that is Euler's force. Anyway, with the help of all these forces, we can actuate the fluid from particular place, this place to this place, even this place to this place. There are different types of oils will be there. From one place to another place, we can actuate the fluid and we can perform the reaction. Even we can do multiple reaction in one platform. Like say, in a chemical reaction, you have 5 mixing steps. So like earlier DNA hybridization, only one mixing step. Now in a biochemical reaction or a chemical reaction, you have 5 mixing steps. Then you can do 5 vessels in that fluidic 1, 2, 3, 4, 5, and that is a valving. One to another, another to another, it will be coming slowly, then reaction will be there. Okay. So that is fundamental operations are valving effect, mixing, and aliquoting. Means with this centrifugal force, as you are telling, if you have a 5 microliter of liquid sample, you want to dispense in 10 or 20 ways. So you can dispense in that when you are giving force, in same way it will dispense each of the oils. Valving effect, one to another to another, they will go valving effect, and then mixing by your Coriolis force, because as your laminar flow, that which is the interface, they will be mixing. So in that way, we can get the benefit of your centrifugal force operation. So here, what you are showing it. So this is your one oil where your sample you are giving. There are different oils actually, and how to design that oil, that depends upon that. First, you cannot do arbitrarily. You cannot do arbitrarily. You put one oil, you cannot do it. First, you have to simulate that whole the things, in which position, what force amount will there, and what will the dimension of each of the channel. These things we can use by your software, that Cosmosol or so many other softwares are there. You have first simulate to design that things basically. In which position, what vessel, what will the dimension, what will be your fluid channel. Because here that your force response, centrifugal force, Coriolis force, then surface tension and viscous drag. All these four forces are responsible, and you have to calculate accordingly, and then you can design that vessel and how that mixing things will be there. So this is that means your sample preparation, means one platform you have to do nothing basically. You have to do nothing, only you give one drop of blood here. Blood means it contains cell and serum. Now if you in certain RPM if you rotate it, then RBC and all cellular material will be separated from your plasma. Then plasma will be actuated from this place to another place, where that vessel contain one biochemical, means your reagent that mix up this. Then another it will be coming, then you will be getting color and get the readout. In that way you can do all the complex reaction in one platform. Even you can do the DNA hybridization here also. So that dimension is same, 1 centimeter by 50, 100 micrometer and 40 micrometer height. So this is the ideal way to show that how that it works actually. Here you are putting the sample, then you are giving one, here it is coming, then ultimately the wastage coming it out. And you are getting each oil, there are some reagents already you have put it earlier, reaction will be coming it out. Now question comes, how will you construct this platform? Simple channel is very easy. You just issue it the normal MEMS platform, you do that channel. But how will you construct this fluidic platform? So here is that, I think this is that how to construct this layer by layer construct. So these are the 1, 2, 3, 4, 5 sheets are there. So if you now if you look for that, the sheet number 1 and sheet number 5, these are the dummy actually. This is the dummy. Actually this is the sheet where that your dimension you have already optimized, or you have by your means your software and your means your other calculations, you have designed that what will be the dimension of your oils and at what distance. Now with the micro machining you can, that polyacrylic sheet you can make that holes basically. Then you can fix up that 4 and 2 are that adhesive layers. Then you fix up layer by layer, then you can get a 3-dimensional picture, or 3-dimensional feature you can get. That is why that 5 layers fabricated by CNS means your center, that is your means your cutting machine basically. By vinyl cutter, top layer 2 and 4 are pressure sensitive adhesive layer, then middle layer containing all the chambers. Means if you layer by layer if you get, then you get all the 3-dimensional feature. Layer by layer means it is one layer is a 2-dimensional feature. You are giving that 4 layers, then you are getting 3-dimensional feature, and height is around at the 40 micrometer or 50 micrometer. That what you want basically, that you can do it. So here that example is that this is say representative channel. So this is one reaction you can do it here. Another reaction you can do it here. Another. In that way, how many channels you can put or reaction feature you can put, you can multiplex it. Like say, at least not theoretically, we are also trying. Say, when clinician or doctors in a sitting, patient are coming, and doctor is confused whether he has a malaria or typhoid or dengue, because symptoms all are high fever. Then if you have a platform like this platform will diagnose dengue, this platform will diagnose your malaria, this will diagnose your say maybe typhoid, etc. Say 5 platform in a one CD disk, you have a 5 platform. give a sample, 1 drop of blood, then they will dispense all the ways, then all the normal biochemical tests are there, and then we will getting readout. Within half an hour, we will get the readout. Means all the 5 tests can be done, then immediately you can tell that, okay, you are malaria positive, or a typhoid positive, or TB positive, and etc. So that is the beauty of that, your CD platform. Then we can multiplex it very easily. And now if you think that everybody has a CD, now that's hearing aid, that basic machine is available to everybody. So if we can construct these type of things, and all the diagnosis can be done even at that grassroots level, village level we can do it. Now if you ask me whether you have done it, not we cannot claim it really we have performed it, but we are at the way. 50% we have already done. Basic things already we have covered basically. Anyway. So this is the real picture. So the earlier picture is the cut and paste things, but this is the real picture, what we are doing in the lab basically. So these are the layer what we have telling. So we have constructed this one, and a real picture is so this is that whole the setup here. So here we are showing that how that actuating, this is your blood sample, it is going to that here, and as picture is not very good, and depending the RPM which rotation you are giving, that velocity will be increasing. That is very simple. And valving effect is there, and valving effect if you want to see it, then how to design these your each and every vessel and connecting diameter of that is your prediction, that is the main factor. And you cannot do arbitrarily. If you do arbitrarily, it will be lost basically. And another problem is that additions. As our technology is not that much, the adhesive tape, that sealing is very good. If it is not very good, leaking will be there. So that problem we are facing basically. Anyway. So this is another way. Say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like say you have a 10 reaction in a biochemical reactions. 10 steps. So you can perform in that way if you can design properly, and you can perform that 10 reaction. You have not to do in a 1 by 1 tube. So you are only giving the optimizing that which RPM you are rotating and which place you are putting, that is your reaction vessel. Each one is one vessel basically in a microliter volume vessels, which contains predetermined reagents. Already you have put it there, and then you do the reactions. And this is one example of your total multiplexing. Means you can do, you count it how many, around 30 might be, 30 reaction you can do in one platform. Maybe 30 different readout, or same readout in 30 different samples, you can do it. And this is very easy to demonstrate, tell it, but lot of calculation, lot of physics, lot of chemistry behind it, and lot of biology is there. Okay, this is just an, it is not working. Anyway, so that is that thing basically, how will you doing? So that explained what we are doing. We are, instead of fabricating whole the CD platform, what we are seeing earlier, here we are putting one slide, on one slide. So this is one slide, which all that micro channels are there. This is another slide, this is another slide, this is another. Means 4 reactions we can perform, because these channels are fabricated in your glass slide, and we are putting the glass slide in CD platform. Earlier what was in the CD platform we designed, all the fluidic channel designed the CD platform itself. So this is this fluidic platform on the glass slide that you are putting in the CD. So just to demonstrate that really CD works in that way. Okay. Now another is your paper fluidics. Paper fluidics, so far if you can earlier slides that acrylic film has some cost. It is not easy to get, not that much costly. It is not that much costly, but you can get it. But still further you can decrease the price or application by your paper fluidics. Paper fluidics means your blotting paper, you know that, blotting paper. So if you dip the blotting paper, fluid will be coming out due to capillary force. But drawback is that they can diffuse all through it. Can we make a micro channel, but here force is your capillary force. You have not to giving any pressure driven or electrokinetic, nothing else. Only capillary force it works. But can we design that fluidic channel in a filter paper? That is the challenge. Challenge is because capillary force already there, because pores are there. Capillary force fluid will be going on. But can we design microfluidic channel in that blotting paper or say any filter paper? So challenge is that say so here we are the same using photolithography and wax printing and inkjet printing. So what you are doing, waxing it totally. Then we are doing photolithography to etch it out that particular part. Then if you give a drop, then it will pass only through that channel, not the diffusing whole place. So this is that picture. So this fluidic channel are means constructed. Now if you put the liquid, you see that liquid is passing through in that way, not that whole way. So that is another way to, very cheap way to construct the fluidic channel, and that capillary force we are utilizing that which is present. But problem is here that all the papers will not be getting same capillary actions. That is the problem. One will be very quick, one is very slow, and that we cannot, means that is not in our hand basically. That is that how that filter paper means how it is prepared basically, that paper and pencil based devices for your fabrication. So maybe I am taking time. Anyway, so next example we are coming that how that, so we discussed that DNA hybridization, then CD platform for your actuation or doing multiplexing reaction. Now we are coming another interesting phenomenon. So how one protein reacts with another protein to get the readouts, like say disease diagnosis. So you are giving one antigen, one antibody, get the reactions. Same phenomenon if we do in test tube and if we do microchannel, what the difference will be getting, and what new reactions or new phenomenon we can discover out of that. So that we are now coming basically. So that important parameters of fluidic reaction channel height, flow rate, reaction time, shear stress, all these coming to the play. Shear is playing the roles also. So here I am coming directly to the reaction basically. Here we are putting on protein like say antigen or say antibody. Now we are flowing that antigen here. Now antigen antibody will react, then that some color will be produced, we are measuring that color. Now question ask, you can ask that why you are doing it, you can do in test tube. Again we coming to the back point. So if we do the test tube, we will be doing taking a lot of time, and we cannot unravel a lot of biophysical features basically. In DNA the dimension is around say maybe whole the DNA molecule maybe around say 100 angstrom or maybe 200 angstrom. But if the dimension is at the nanometer level, say 1 nanometer or 4 nanometer, then what will be the biophysical phenomenon? That we cannot discover by normal test tube way. And even if we can decrease the dimension in nanometer level, then we can discover what is happening. Okay. I think I shall come directly to that phenomenon basically. Here that experiment is, we are putting say maybe one, forget that actual picture, I am coming for your general audience, say antigen and antibody, two are reacting together and then reaction will be coming. Now as you are telling that, as you are giving fluid, some shear stress is there. Now we are asking the question, what is the optimum shear stress is responsible for getting that highest readout? Highest readout means you are getting the signal will be maximum, signal will be getting when, what will the shear force means, what for it will be giving that your material or that antigen by getting the maximum activity. So here that experiment is that we have increased that channel height. Three features are there: length, breadth and height of the channel. And if we fix the length and breadth, then height we are increasing, decreasing, then your confinement effect you can means visualize. Means if we decrease the height length, your confinement will be more. Confinement will be more. So that effect we are seeing it. We have a height 40 micrometer, 50 micrometer, 70 micrometer, and these are the all that solid lines or dotted lines are simulated lines, and that all that these round or square or triangle shaped are your experimental figures. Now if you see that the 40, 50, 70, when the height is increasing, height is increasing, then the readout is little bit decreasing. At 40 micron is a little bit depth, your color is very high, intense, and it is a little bit less. And if you do the same reaction in open system, there is no confinement effect, you will be seeing the readout is this. So that DNA hybridization already I have mentioned. So this you can think that what is confined effect means basically. And here is that time at 20 minutes or say 30 minutes you are getting that plateau basically, getting the maximum height. And these are the simulated figures. So to decrease the channel height leads the yielding a higher saturation concentration, and that cone bond means say antigen, and it takes 20 minutes. Now question arises here, what are the real effects response will get that highest readout at the 40 micrometer height? So what is happening in the biochemical level? Because all that molecular dimension is around say 4 nanometer. One molecule is sitting in that bottom is 4 nanometer, and another molecule is flowing at that dimension is 4 nanometer. And channel dimension is say 40 micrometer. You can decrease the channel further also, but that facility we have not. Anyway, so now you are asking that what is happening to that phenomenon. Here you can think that all that biochemical reaction, if you think that A plus B equal to AB, that depends upon that forward rate constant and backward rate constant. So in that shear space, both the reaction rate constants are responsible, or one is responsible. So we simulate all these things. So we are describing a 3 model with a model 1 where that shear rate is invariant and Ka and Kd are changing, means your association and dissociation constant are changing. Second model we are assuming Ka is invariant, Kd is constant, and model 3 both K and Kd. So with these 3 models we simulate and get that this is the simulated figure, these are the simulated figure, and this is your third model. Now if you do the experiment, experimental model is fitting with that third model, means here we are seeing that with this micro confinement and that particular shear rate, your reaction rate is maximum. When wall shear rate is 0.5, you are getting maximum readout, then slowly is decreasing, and here both K and Kd means forward and backward reaction, they are influencing. That phenomenon, it can be discovered only by that microfluidic situation. You cannot do that means observation with the open system. Open system you are getting that you cannot control anything basically. So after particular shear rate, that reaction readout is decreasing. So why it is decreasing? You cannot do in open system. Decreasing, here we are predicting that as you are giving a more shear rate, that molecular interaction after that binding, it is breaking the bond basically. That we have to establish. We have not yet predicting basically from that experimental phenomenon that is happening. Like say 2 molecules are coming, they are coming closer to their particular shear rate. When they are increasing shear rate further, they are going apart. That physically we have to establish that, but this is the observation, then we have to establish. And interesting phenomenon is that that is true for all the flow rates in all the your fluidic means your confinement effect. So that effect we have discovered due to the advent of your fluidic platform. Otherwise, it is impossible things. Okay, so with the we now capitalizing this platform for your diagnostic purposes. Diagnostic purposes like say in cancerous situation, cellular surface, that architecture is changing, that glycosylation pattern, maybe these are the very wrong, you are not familiar with these things. Anyway, some molecule, some pattern is changing. Now can we utilize this platform for diagnosis purposes? So here that is that normal that flow chart, you take the sample, prepare the microarray and statistical analysis, data validation, that is you forget it. So we have that arrays of say 1, 2, 3, 4, 5, at least 20 molecules or 20 lectins, say arrays of lectins. These are the array we have make it in a platform. Then you are giving the sample, and ultimately we are getting that readouts. Some are getting means reds means your interaction is high, green, and this means there is no interaction, is 20. So you take the blood sample, and with that array we sample passes the sample with that flow rate, and we get the readouts. These are the normal sample, These are the gastritis, adenocarcinoma and type C. So you have not to go in detail. What we are seeing it here, say 50 samples we are taking from 50 patients, then within a 1 hour we can finish up all the reactions to get the readout. Then we are analyzing using that is your software, that is your image analysis, that which pixel density is high or low, then we are getting. And interestingly, this result, what we are getting from this fluidic platform, this result, it is telling that what doctors are saying. And if we can do that experiment in a normal biochemical lab, it takes 2 years, even lot of standardization necessary in column, lectin, that is huge amount of expenditure. But we can miniaturize all the things in a one platform. I am not claiming in one platform, but individual reaction, but we can do in one platform, and we can finish up the reaction in 1 day. So that is the achievement you can tell that. So microfluidic platform you can do when sample is very less. You cannot get sample like 10 ml blood. Here you are getting sample maybe few milligram of your biopsy, few milligram, 2 milligram, 3 milligram. Sample is limited, and we have to read out that whether interacts with 20 molecules or not, and in that we can finish up 50 to 100 samples in a very limited time. So that is, forget these are all the things, we can analyze all the things. We are coming to the next step, last topic actually, that is fluidic behavior at the single cell level. As I have mentioned in the beginning of the lecture, that cell dimension is 10 micrometer. If you want to analyze that feature at the single cell level, you have to think about at 10 micrometer level what is happening to the cell. What is happening to the cell? Now, if you look for the cell, inside the cell is a liquid. It is a colloidal solution. In a cell, inside the cell it is a colloidal, not an rigid, not an totally transparent. Now, question arises, how that fluidity, that molecule transport from one place to another to get the nutrients and all that, or you can tell that information, how that information passes from one place to another, because it is a highly viscous liquid and it is a malleable. What force is going onto the surface to change its phenomenon basically. So here that this is the cell, it is dimension is around 10 micrometer. It binds your extracellular matrix at the same time with other cells. Now with this fluidic platform, we can measure this force, at which force cell attached to that surface. I think you can think that how will you measure it? We know that only one, if we drag it or force it, we can know how much Newton on it. In the cell level, one cell dimension is 10 micrometer. It is attached to the surface with some molecules. How we can measure this force? Now, if we go through, we flow the fluid in that way, what happens to that phenomenon? How that physical force coming to biochemical phenomenon and get the biochemical level. So that is the main thing it here. So what is happening it here? Same way PDMS channel and everything we have here. This is your cellular analysis through the traction force microscopy based on biomicrofluidics. Traction force means when that this is a cell, it is sitting in the surface, but it is not, it is adhering actually. So what traction force it is adhering to the surface, we are measuring by microfluidic platform. Means like say we have a four legs with the attached it here. Now if you give some pressure is here, then it will be distorted. Now in that way we are measuring, but how will you measure it? For a macromolecule we can do it, or macro things, but a small things you cannot visualize it. How will you do it? We are doing it indirectly. What is happening in the platform? We are giving micrometer level fluorescent beads. That is a soft miniature platform. When it is attached, we take photograph the cell. Then we are giving flow. Then as it is a moving, then the displacement of that surrounding happens. Then all the beads will displace. With this displacement, we can calculate that what force is imparted. So in that way, we can measure the traction force. And traction force, and it is different for your different types of cell, that behavior. So this is the whole platform basically. That cell is here, we are giving the flow, then we are, these are the beads basically. Then we are taking the photograph and we are measuring that what is happening. I think these are all the things. So here that is your traction force landscape. Means this is one cell and these are the places where that really you can tell that these are the legs of that your cell, like your pseudopod basically, where that cells are attached, and these are the traction force your landscape, and these are the micronewton level, that force micronewton level it attaches. Now different cell has a different types of attachmenting force, and that force is, that physical force is going to your biochemical reactions. Same thing in blood flow. I am coming to you. Now here is that what is a micro confinement effect. In a cell in your body, in the capillary, it is a micro confinement effect, because all that cellular hormones and everything that is not diffusing throughout that things. So that is why you are getting that micro confinement effect. Influence of micro confinement. So chemical, due to the restricted diffusion, confinement increases the concentration of the releasing growth factors. And the physical things, cell reduces the effective channel height, hence the stress experienced on cell membrane is larger in micro confinement than in large conduits. Means, say in a cancerous cell, what is happening? You know that metastasis possibly will, you have hard possibly in one place cell to going to other place. But it is a how that cell, cancer cell, withstand that much shear of your fluid flow. Generally all cell will die, but cancer cell you not dying. So that means they have that membrane behavior in such a that they can withstand that much of shear. That is why, so they can transfer from one place to another place actually. So we have discovered that we can standardize that traction for microscopy. So these are the fluorescent beads what I am telling you. We take the picture, then we are giving flow, then it will be bending will be there, then movement of the beads will be there. Then from the movement you can measure the traction force. Okay. So confinement effects here is that event one, if we apply flow shear stress on cells, they first rearrange their adhesive pods, means that traction force place that is your landscape of the traction force. After that event, say it takes might be 5 seconds. After that 5 seconds, that force that changes to focal adhesion and then converted to biochemical signal that propagates through cytoplasm, act membrane, increase the fluidity. This is only one readout. You are giving force, then you are measuring the fluidity. In between these two times, we are telling it is a response time. We have coined that time is a response time. Now we are asking the question, response time is different for your different cancer cell and normal cell. All cells are visually same, 10 micrometer diameter, they have a membrane at that macro level. But if you go through more details, cancer cell, different types of cancer cell behave differently and normal cell behave totally differently. Now you are asking, this response time, is it different? Because we know that phenomenon, cancer cell can traverse from one place to another without any damage. So we have find that really they are different. The response time is greater, less for cancer cell which are metastatic cell, but which are normal cell, this response time is very high. Means we can conclude that with that fluidic platform, we can discover these biophysical cues or biophysical, you can tell the readout, by which we can tell that metastatic state of the cell. Means response between physical force to chemical force, like say fluidity of the cell. So that is another phenomenon can be discovered or can be observed only utilizing that fluidic platform, microfluidic platform. So we are concluding here, cancer cell may be more active within the micro confinements of tissue than what observed in open culture. Open culture means open system. So this is the all basically. So basically I covered that, or what I don't know how much I can convey to you, but what is microfluidics, particularly in biological perspective, and different aspects of fluidics starting from diagnostic to cell biology, even in basic fundamental questions I have discussed. And This is not a single person job. Basically, involved Professor Suman Chakraborty, IIT Kharagpur Mechanical Engineering Department. His lab basically develops the fluidic platform and everything. Then Mark Madhu, University of California, for your CD based things. Then Professor Gautam Chatterjee from Kolkata Medical College, he is providing all that biological samples for the analysis. And these are the students actually, Vivas Roy, Kamal Das, Devapriya Chakraborty, who are involved in that work. And you are getting fund from DVT, DST, NS7, etc. Okay. So this is the only a glimpse of that microfluidics actually. There are a lot of revolution is coming, development of biomicrofluidics. One journal is devoted to biomicrofluidics and lab on HEP is there. So we are far away what European country and American are doing, but we are in the race basically. Thank you. So if you have any question or any query, if you can ask. I try my best to answer you. Yes.
How you are rotating your CD?
That is centrifugal force, just centrifugal force.
Capacitor actuator?
Yeah, capacitor actuator.
And regarding the paper microfluidics,
Yeah.
then which paper you are using?
Generally, we are using your that is hot band filter paper. We are using hot band filter paper. But problem is paper fluidics is that standardization. You cannot get uniform capillaries always. We are assuming that hot band paper is more or less is uniform. That is that problem actually.
After putting a wax on it, then after you're doing a photolithography, you're able to control the capillary effect.
No, we cannot control the capillary effect. It is not in our control. Only we can direct it. Direction that we can control.
So you're putting a wax on the top and the bottom.
Top, bottom, both. CD. That is one CD platform is 10 rupees. That is our calculation actually. One CD platform, all that 4 layers, it is means if it is coming to the plate 10 rupees. Disposable or use and throw. You cannot use, reuse. You cannot use, use and throw. Leak proof, that is adhesive layer one I am telling you. That is a problem actually. That adhesive layer is not available in India. We are getting some from some Mark Madhu's lab, and sometimes it works, sometimes it is not working. That is another problem.
After applying adhesive layer, we
give another layer. Then you are getting all the 3-dimensional feature there. From 2-dimensional, you are getting 3-dimensional. In the normal your fluidic platform, that is channel, you are giving a glass slide, then your PDMS layer you are giving. So you are getting a 3-dimensional feature. Here we are not using PDMS, we are giving a layer by layer, 3 layers, and getting that your feature.
So you told about different forces
Yeah.
to actuate the
Yeah.
if it is applied for mixing of two fluids,
Yeah.
whether we can control these forces to get the uniform mixing?
No, that is the problem of your laminar channel or single channel mixing. I am telling that it is a blue liquid, is a red liquid. If you pass through it, you will be seeing that blue red, they are passing parallelly. Only that interface zone they are mixing. To mixing that blue and red, if you want to mix it, then we are using parallelly electroosmosis. That serpentine channel, and that this is your fluid direction, you are giving electroosmosis in that direction. By which mixing will be there. That is that you can tell that phenomenon basically. Drawback. In a laminar flow, mixing is restricted. But if you have turbulent flow, then mixing is there. So you are making turbulent by using serpentine channel and electroosmosis. Or there is some flap, lot of other things are there. But we can capitalize this non-mixing thing also. That is other, a lot of other things are there. What is it? Acoustic. Yeah, yeah. acoustic can be done. Vortexing. Vortexing can be done. That is not a micro channel. You can make a micro oil basically. Micro oil, there vortexing you can do by magnetic bead or nanoparticle bead. So that is another area where that people are working fluidics for your micro reactors. Maybe 500 microliter reactors or mixing is very much necessary. There vortexing is very much necessary. And vortexing used for your cell patterning, lot of other things are there. I am telling is only a part. We are doing a lot of other things. Here you are getting only a part things we are telling. Yeah, surface texture can be changed. Particularly for cell biology things, say surface texture if you can change, you can modulate. How much wedge ridges you want, what feature you want, that will restrict your cell adhesion and cell behavior. That feature you can do it also. That pillars, how much pillar density, what the pillar length, etc.
Sir, sir, when you're putting a wax on both the up and bottom of the microfilm, so when you put a drop on it, so what about the lateral diffusion that is taking place?
No, that is why axial diffusion after that waxing, then you are giving photolithography, removing that wax. Then if you give one drop, the lateral diffusion is there. If you go to the picture, you will be seeing the lateral diffusion is there, but it is not nil. So that is one of the means you can take a drawback actually. So how much when you are actuating, lateral diffusion is there also. That you cannot make it nil actually. That is the drawback of paper plate. But it is easy to construct and cheap actually, use and throw. There is a, if you CD disc cost 10 rupees, it will cost maybe 50 paisa. Okay? Hmm. Hmm. No, one thing is fixed. No, probe is not fixed. Probe is moving. Probe is moving. Probe is, it is ligated with some fluorescence. Probe is moving. That complementary stand, that is your binding to the surface. No, sample testing, ideally what will happen, you isolate the DNA. In the fluidic platform itself, you can isolate the DNA. From the cell you break, isolate the DNA, you cut it. Then with the heat, 50 degrees centigrade, you can DNA, make it single stranded. Make a single stranded. So that is your, maybe you are diagonizing that sample. Oh, in that way you are telling. In that case, we have the probe should be in your bottom, will be fixed in that case. Okay.
Thank you, Professor T.K. Maiti, for a wonderful talk. Now I would request Professor Somendas to kindly hand over a small token to sir. Thank you. We will now have a small break for tea and then reassemble here.